we have got codon on the mRNA, and complementary to which anticodon is present on the tRNA. Okay, and it will attach and it will produce protein synthesis, right? But not what we can do is, we have got a group of drug known as aminoglycosides. They will produce misreading of mRNA. Okay, so this misreading of mRNA is done by aminoglycoside. So basically what happens over here is, just suppose we have got AUG as the code on the mRNA at A site, so that tRNA will attach, which contains the complementary codon as UAC, right? No other tRNA can attach. So this will carry one particular amino acid only. When we give aminoglycosides, this code is read wrongly, means instead of A in AUC, it can be read as C. So when it is being read C, a wrong tRNA comes. This will contain a different anticodon. So when a wrong tRNA comes, it will carry a wrong amino acid. That means protein which we want to synthesize will not be synthesized, but rather a wrong protein is synthesized, and most of the time this wrong protein is a cidal protein, and it will kill the bacteria. So this is very much important to remember, that aminoglycosides are the only protein synthesis inhibiting drug which are cidal. Like most of the other protein synthesis inhibitors, they are bacteriostatic, but aminoglycosides, they will actually kill the bacteria. Okay, drugs coming under the category of aminoglycosides are streptomycin, gentamicin, tobramycin, natilomycin, neomycin, capreomycin, kanamycin, and amikacin. Now, as a group, special point to remember about aminoglycosides are not effective orally. The reason is that they are not well absorbed. Now, because they are not effective orally, we need to give aminoglycoside by injections only. Next, their aminoglycosides are mainly effective against gram-negative bacteria. Like if you compare penicillins, they are effective against gram-positive bacteria, right? And these aminoglycosides, they are basically effective against gram-negative bacteria. Even they are also effective against pseudomonas also. Next, aminoglycosides are not effective against anaerobic bacteria. Now what might be the reason is, basically the aminoglycoside, they require oxygen. So any bacteria that uses oxygen, means whatever aerobic bacteria are there, via oxygen the aminoglycosides can enter into the bacteria and they can inhibit the bacteria. But in case of anaerobic bacteria, since they do not take oxygen, so aminoglycoside, they cannot enter inside the bacteria. So therefore, because of this reason, aminoglycosides are not effective against anaerobic bacteria. Next, aminoglycosides are cidal drugs. These are the only protein synthesis inhibitors which are cidal, and all other drugs in this category, that is protein synthesis inhibitors, are bacteriostatic. And the reason we know, because aminoglycoside will produce cidal protein, there will be misreading, because of which a wrong protein will be produced, and that protein is usually a cidal protein. So therefore aminoglycosides are the bactericidal drugs. Next, special side effects of aminoglycosides are, they produces ototoxicity and nephrotoxicity. Even they are producing neuromuscular blockade. Now among ototoxicity, they are of two types. Like we have got auditory ototoxicity as well as vestibular ototoxicity. Now over here, amikacin can cause maximum auditory toxicity, that is auditory ototoxicity, whereas streptomycin can cause maximum vestibular ototoxicity. Now vestibular system is to maintain the stability of a person, so therefore this streptomycin will affect the stability of the person. Lastly, nephrotoxicity is produced by neomycin. Okay, and neomycin not only it will produce nephrotoxicity, plus it will also produce neuromuscular blockade. Okay, so this is all about aminoglycosides, and this was all about my today's video regarding protein synthesis inhibitors, the antimicrobial that will produce action by inhibiting the protein synthesis of the bacteria. I hope you people found this video helpful. Thank you for watching.